Today, we are taking a deep dive into Hericium erinaceus. You probably know it as lion's mane, or maybe the beard-tooth fungus. Visually, it is pretty distinct. It looks more like a cascading waterfall of white icicles than a typical mushroom.
Yeah, and culinary circles love it because it genuinely tastes a bit like crab or rock cod. But our mission today is, well, we are moving way past the dinner plate.
Right, we are digging into a stack of research all about neurohealth. We've got chemical analyses, bioavailability studies, and some really interesting data on memory enhancement.
Exactly. And usually when we talk about medicinal mushrooms, the science feels a bit vague, right? But the sources we are looking at isolate very specific bioactive components.
Yeah, primarily found in the mycelium, the root structure, rather than just the fruiting body we actually eat.
That distinction is crucial. The literature really zeroes in on two compounds, Erinacine U and Hericine A.
Let's look at erinacine U first. It is described as a cyethane diterpenoid, but the functional takeaway is its impact on neurite outgrowth.
Right, so think of it like a construction crew for your neurons. For your brain cells to communicate properly, they have to physically branch out and connect.
And erinacine U essentially stimulates that branching process,
It does.
which is almost ironic. The mushroom itself grows these long dangling spines to interact with its environment, and its compounds basically force our neurons to do the exact same thing.
To reach out and connect, yeah. But simply growing branches isn't enough if the signal doesn't get through. That is where Hericine A comes into the picture.
And this was probably the most surprising part of the research for me.
Oh, absolutely. Because normally, to trigger nerve growth or survival, you need neurotrophins like nerve growth factor-NGF, and they operate on a really strict lock and key system.
Right, where the lock is a receptor called TRKB, and usually if you do not have the specific key, that door stays shut tight.
Precisely. But Hericine A kind of cheats. It activates the ERK12 signaling pathway, which we know is vital for memory, but it does it entirely independent of that TRKB receptor.
So it's essentially a biological backdoor.
That is the best way to look at it. It creates a pan-neurotrophic effect without needing that standard key.
And the results in the animal models were pretty stark.
Very. Mice fed just 5 milligrams per kilogram, which is a tiny dose, showed not just higher levels of brain-derived neurotrophic factor, but significantly enhanced recognition memory.
But I have to play skeptic for a second here. We see this all the time, right? Amazing mechanisms in a test tube or a mouse, but it totally fails in humans because of the blood-brain barrier.
The ultimate gatekeeper.
Exactly. It is designed to keep foreign substances out of our gray matter.
It is, and
Yeah.
that barrier is where most neuropharmaceuticals just hit a wall and fail. But we have bioavailability studies here specifically for Erinacine A that actually challenge that norm.
The data suggests it crosses via passive diffusion, right?
Yes, meaning it doesn't even need a transporter. It essentially just ghosts right through the barrier.
Which is wild. And the studies showed it appearing in the brain as early as 1 hour after ingestion.
And it persists. The peak concentration wasn't until 8 hours later.
That duration is massive. But also, looking at the data, the unbound ratio is close to unity.
Which is a huge detail.
Right, so for any non-chemist listening, that basically implies the compound isn't getting tied up by proteins in the blood. It is flowing freely between the blood and the brain, so it is actually available for the brain to use.
Exactly. It is not just getting in. It is staying active and doing its job.
So to pull this all together for you listening, we aren't just looking at a seafood substitute anymore.
Not at all. We have a fungus containing compounds that stimulate physical neural branching.
And they use a chemical backdoor to activate memory pathways without needing the standard receptors.
Plus, they can passively walk right through the blood-brain barrier and just hang out for 8 hours. It really bridges the gap between folklore and molecular biology in a way we rarely get to see.
It definitely makes you look at that old log in the woods a little differently. Because if Hericine A works by bypassing the standard locks on our nerve cells, you have to wonder what other biological backdoors are hiding in the fungal kingdom that we haven't even found the key for yet. Thanks for joining us in this deep dive.